Okay, thank you for organizing this event. I am Dipanjan, a senior postdoc in Andrea Schaefer's lab, and we in general work in the lab on the field of olfaction. We are interested in understanding how the temporal structure in odors are encoded and perceived in mammals. So in the natural world, odors get diffused naturally, as you can see in here. And if we stick a fast detector, like the one from here from Aurora Scientific and Mini PID, what we get is a rapid fluctuation of concentrations of odor. So an important question lies is that what information does a naturally occurring odor plume carry? This is what we are trying to understand from this project. To do that, we use two of these mini PITs fitted with two different UV lamps, a 10.6 and an 8.4 lamp, and now presenting odors in two different configurations. In odor 1, we had a separate odor than odor 2, and we presented them as a mixture or as separated sources. Now, to create a naturally turbulent environment, we used a fan and an obstacle to create almost an outdoor kind of environment, and hence observed that the indoor odor plume recordings were very similar to the outdoor ones. Now, when we presented odors from a single source or as a mixture, firstly, what we observed was the rich temporal dynamics, as you can see in these raw traces. Further, what we observed was that the odor plumes occurring from the two different odors were highly correlated among themselves. Further, when the odors were separated by as small as 50 centimeters apart, the plumes seemed to be quite uncorrelated, rather well uncorrelated, as you can see from this plot. So we estimated the correlation coefficient for the different cases by separating the odors in tens of centimeters. And what we observed that indeed the correlation coefficient followed a decline monotonically from a mixture or a single source to two sources being just 50 centimeters apart. This suggested to us that maybe correlation coefficient might be an important cue to understand odor source separation. We went further and used different odors to test the same thing and did that for different radial distances of 20 or 60 centimeters, and as well as in outdoor conditions. In all the cases, what came out loud and clear was that correlation coefficient between odor plumes might be an important way to understand what is the difference between two odor sources in spatial context. So we wanted to ask if this is indeed a cue that is used by any of the olfactory driven animals or mammals. So we went to the literature and observed that in insect world, indeed the temporal structure in odors has been seen to be perceived and encoded in different stages of the olfactory system. For example, in all these papers in the recent past. However, in the mammals, this was largely unknown. So we went further to try to ask whether mice can understand correlation structure in odor cues. So to do that, firstly, we wanted to build an odor delivery device which could reliably deliver odors at different frequencies without disrupting the temporal structure. And we used these high-speed valves from the company to build this. And what we get is that the odor delivery device could deliver odors at different frequencies up to 80-100 hertz without much disturbance in their temporal structure or amount of odor released. Now, having this device built, we created two odor stimuli, a correlated and an anti-correlated odor stimuli, which just differs in the phase of when the two odors appear. So in the correlated case, the two odors appear in phase while the anticorrelated they are out of phase. We also used a different set of valves to control for the flow so that the net amount of odor or the air released during the process is constant for the two cases. Now that we have these two stimuli, we fitted in to our home-built self-operating operant conditioning system, what we call as the autonomous. In this, animals live happily in an enriched environment with free food. However, for water, they need to run upstairs through this ladder and come into the second floor. In here, they would need to get into this tunnel, and we have a really strict door policy using a magnetic toe so that there is only one animal at a time doing a task. And to get water, they would need to do a task in this end of the tunnel. Now, each of these animals have a RFID chip attached underneath the skin and the neck, which helps us identify a single individual animal. Now, when they are there, they are given to identify whether a given stimuli is a correlated or an anti-correlated stimulus, using our well-described TOD as before. Now, just to recapitulate the task, half of the population of the animals were given to are asked to identify if a stimuli is correlated, and they would get a water reward for this, while the other half is given a water reward for the anti-correlated trials. And what we see from our star animal is that, indeed, after a few hundreds of trials, the animal could understand way above chance the difference between a correlated from an anti-correlated stimuli. And this he could do even good way above their sniff frequency, that is around 16, 20, 25 hertz of odor delivery, which makes the task difficult. Now we did multiple controls. One of the important ones was a scrambling of the valves. So we used a set of valves to train the animals and then scrambled it around to check if the valve clicking is not creating a different set of cues for the animals. And what we see is that even after the switch control of the valves, the animals perform very similarly, as you can see in this plots. Here are some other examples from different animals or in the cohort. And what we see is that indeed the animals, all of them, could perform way above chance at lower frequencies, discriminating correlated from anti-correlated stimuli. However, as we are increasing the frequency of the task, that is making the task more and more difficult, the performance starts declining. But it's still way of chance at around 20 hertz or 25 hertz, which is way beyond their sniff frequency. Looking at the entire cohort, a amount of around 1 million trials, what we see is that the same picture. That is, animals can discriminate between correlated and anti-correlated trials. However, the control animals who are not given a perfect cue to do the task could not do at any point of time. We tested this with different odor pairs and also using a correlated versus uncorrelated trials at 10 hertz. All of them pointed out to the same picture, that mice could understand correlation structure in general. Next, we wanted to understand how does this behavior get encoded in the mouse brain. To do that, we did some in vivo interrogation in the olfactory bulb, the first area of olfactory sensory system being encoded. We used 2-photon calcium imaging and in vivo whole cell electrophysiology to do it. What we get from the 2-photon imaging is that we use Tibet-Cree GCAM6 mouse for this preparation. So the projection neurons in the olfactory bulb turn green, helps us understand the calcium encoded in these neurons and hence the excitability in these neurons. We present 20 hertz correlated and anti-correlated stimuli in these animals and then try to understand how the integral or the amount of activity in these neurons for the specific stimuli helps us understand the difference between the two stimuli. What we see from these examples is that a tens of neurons in the bulb could encode the correlated and the anti-correlated in a different fashion, as you can see in these cases. This happens both in the anesthetized and the awake conditions. Now, when we present the integral to a linear classifier, we observe that just a few hundred of ROIs could help the accuracy of the linear classifier reach its around 80-85%, suggesting that it may be a work of a few hundred or a few hundreds of cells to encode the entire phenomena of to understand help understand the mouse a correlated from an anti-correlated odor stimuli. This happens the same when we even give them finer bins of data. Now, for the whole cell Paxclamp recordings, what we get is that we present frequencies from 2 Hz to up to 20 Hz and get the raw traces for an odor stimuli and for a blank mineral stimuli. Next, we extract the subthreshold membrane potential from these traces and cross-correlated them with the PID or the main odor signal that we are presenting to the animal. Now we take the peak of this cross-correlation for the odor and the blank case and normalize them to estimate a matrix called as coupling coefficient or CPC. So a high CPC means that the cell can follow a specific frequency, while a low one means that it does not follow it. Now what we get is that a subset of cells can indeed follow the frequency well at 2 Hz as well as 20 Hz, and this happens way above the significant threshold from the shuffle controls, indicating that neurons in the olfactory bulb can actually follow odor stimuli at different frequencies. Now, when we presented the correlated and anti-correlated stimuli onto the animals while recording from these from the olfactory bulb neurons, we observed that a subset of neurons can indeed encode the correlated and the anticorrelated in a different fashion, both in terms of spikes, that is using PSTHs, or in the subthreshold regime. Now, when we compute the number of spikes in the first 500 milliseconds and the change in membrane potential, we observe that indeed the subset of neurons were different for the two conditions. However, we need to keep in mind that a majority of the neurons did respond very similarly for the two conditions. But based on our calcium imaging data, only a subset of neurons might be important to make the animal perceive the difference between the two stimuli. So overall, to conclude, what we see from our interrogation is that naturally occurring odor has a rich temporal dynamics. Chemicals that come from the same source is highly correlated, while from different sources are uncorrelated or completely anti-correlated. We use microdisperse high-speed valves with which we can deliver odors at very high frequency, up to 50 Hz, while when we present these correlated or anti-correlated stimuli to mice, we see that mice can actually understand correlated from anti-correlated stimuli beyond the sniff frequency, around at least 20-25 Hz. Further, we see that the correlation structure is represented in the output of the olfactory bulb using 2-photon imaging and whole cell recordings. And altogether, what we'd like to convey is that mammalian olfaction is a high bandwidth sense, which is something which is a large unknown in the field so far. I would like to thank Andreas Schaefer, our supervisor of the lab, who actually conceived the project, and Tobias Eccles and Andrew Erskine, who are equal contributors in the project, and the rest of the lab. And I would like to thank the CRIC, UCL, and the Wellcome Trust for the funding. Thank you very much.